Hello, third year, and welcome to the Carry On Chromosome section on genetic engineering. What we intend to do throughout these slides is just like what we have done before with the inheritance ones, is we're going to try and just take you through the key concepts to do with genetic engineering, highlighting kind of keywords, definitions, and taking you through the process of genetic engineering in the hope that it gives you a wee bit more of a steer when you're completing some of the tasks online. So here we go. So one of the first things that we can do for genetic engineering is we can actually look at the definition of what genetic engineering actually means. And your teacher will probably ask you to view these clips. They are very short, they're kind of short and snappy, to the point about what genetic engineering is. So what you can do is, again, just follow the hyperlinks, click on them, and hopefully you won't have to wait too long for them to pop up. So what is genetic engineering? The top bullet point there is your typical definition of what genetic engineering is. So it is the process in which scientists have managed to transfer DNA, genetic information, from one organism, where I'm giving the example, e.g. a human, to another organism, for example, a bacterium. So that's the key thing, is we are taking genetic information from one organism and we're placing it into another organism. So the second bullet point down there is talking about, well, what is the name that is given to an organism who has had its genetic material altered. So the organism whose genetic makeup which has been altered is said to be reprogrammed. You have changed the makeup of the TV. It's a wee bit like auto-tuning your telly. So if you get a new channel, for instance, you have to reset your channels. You are reprogramming your TV to what it was before. In terms of when you have an organism that has had its DNA altered, we talk about that organism being genetically modified because it has had its genetic material changed. Okay, so the first slide there, dealing with the definition of genetic engineering, make sure you know that, and looking at, well, if you alter the genetic information of an organism, we talk about that organism being a genetically modified organism, or a GMO for short. Then you will be able to, or you need to be able to identify what useful substances have actually been genetically engineered. One of those you have come across before in SMS. You have talked quite extensively about insulin and how diabetics rely on type 1 diabetics rely on insulin in order to regulate their blood sugar levels. So insulin is a substance that has been genetically engineered, as has growth hormone and something called factor VIII. So being able to identify at least one substance that has been genetically engineered, we'd be happy with that. So just pick one and remember it. And I think for most, they often remember insulin because it is something that they have studied in previous topics. So what is insulin? Insulin is a hormone that controls blood sugar levels and is given to diabetics. So again, remember we have talked a lot about insulin, so you could, this is sort of recapping right the way back to SMS when you were talking about hormonal control and diabetes. There is another hormone. Growth hormone is a hormone secreted by a gland in your brain called the pituitary gland, and it is important in the growth and development of individuals. You've got something called factor VIII, that is an important blood clotting protein, and it is given to people who suffer from haemophilia. If haemophilia is, you've not heard of that, why don't you look it up and broaden your understanding and knowledge of biology. So what we're going to start off with when we start looking at genetic engineering and the process of genetic engineering is we are going to be looking at what microorganisms do you transfer genetic material into. And a lot of the time we transfer genetic material into microorganisms, in particular bacteria and yeast. So bacterial cells are often used in genetic engineering. And we've got to think, well, why? What is so special about bacteria that they get their genetic material modified? And it's a really simple reason. Bacteria are able to grow very quickly and they're pretty cheap. So that's why scientists will often use these types of microorganisms in their studies, because they can get results very quickly at a very low cost. So genetically modified bacteria can multiply at a very rapid rate, and you can actually manufacture large quantities of useful products very, very quickly. I've got there as well that other microorganisms such as yeast can also be used in the genetic engineering process. So this is hopefully a recap way back to topic 1 when you started looking at the structure of bacterial cells. If you can't remember the structure of a bacterial cell, then it is definitely worth going back and just giving yourself a wee refresher of what bacterial cells look like. For instance, you are going to have to know, and we're going to talk a lot about this section of, or this part of a bacterial cell when we talk about the process of genetic engineering. So bacterial cells do contain plasmids. There is no true nucleus. We have got circular chromosomes, and we also have this plasmid. So we are going to be talking about the stages of genetic engineering, and as a wee sort of lead into the stages involved, there is a wee clip there. It's a couple of minutes long, and it is taken from National 5 Bite Size. So I would like you to watch that wee clip before we start looking at the different stages. So this diagram, it does look quite sort of, quite big and lots going on, and you're right, there is quite a lot going on. But what I would like you to take from this diagram is looking at the different stages and what is happening at each stage of genetic engineering. So the example that I've used here is we are trying to make insulin from a bacterial cell. Bear in mind that insulin for us, if you're a diabetic, you need insulin to regulate your blood sugar levels. So the first stage in genetic engineering is you have got your animal cell there, and inside, I'm just going to use my wee pen here, inside the nucleus, so that's the nucleus here, okay, that's the blue bit, you will find your chromosomes. So your chromosome is this section here, okay, and this wee black bit is the gene that we would like to take out of that chromosome in order to put into a bacterial cell. So the first thing we need to do is we need to identify the chromosome. We need to then identify the gene, And what we need to do is we need to cut that gene out. So we need to get, I'm just going to get my wee scissors here. So we're going to cut this gene out using our biological scissors. And those biological scissors are called enzymes. So to cut this insulin gene out, so this bit here, we need special enzymes. Okay, so that's kind of the sort of human or animal side of things, is we're taking genetic information from that organism, the human for instance, and we want to take that genetic information and put it into another organism. And in this case, we are looking at bacteria. Okay, so this is our bacterial cell here, very similar to the one that we've got on the slide previous. And what we're interested in is this plasmid. Okay, so the plasmid is going to act as a little link between the animal cell and the bacterial cell. So we first must extract the plasmid. So we need to extract that plasmid, and again we need our special scissors, and we need to cut the plasmid open. So we have our plasmid, which we need to extract. Sorry, I don't know if you're aware, I had to pause my filming there because my wee boy needed a pee. So sorry about that. So we've got our plasmid, and we need to then cut it open. So we're making, if you see here, we are making a space for this insulin gene. So both, in order to cut the gene out and to extract, well, once we've extracted the plasmid, we then need to cut the plasmid open. So the insulin gene, which is the black bit there, okay, and that's the gene that we want our insulin from. The gene is inserted into the plasmid and they are sealed together. So you've got your plasmid and you have got your gene, and the two of them get sealed together. And that sealing is done by another type of enzyme. So we've got two different types of enzymes, one to do the cutting and one to do the sealing. Okay, so anything to do with genetic engineering, if you're cutting anything out or if you're sticking or sealing things together, it's to do with enzymes that carry that process out. So once we have got our modified plasmid, so that's our modified plasmid here, this bit here, we then insert that back into a bacterial cell. So we now talk about that bacterial cell being genetically modified, or it has been reprogrammed. And once it has been reprogrammed or genetically modified, we know that bacterial cells reproduce very, very quickly. And what happens very quick is one cell becomes two, and again, you've maybe clocked that I've had to pause again because I keep getting interrupted by little people. So one cell becomes two, and two becomes four, four becomes eight, etc. So over a period of time, you do find that you have a large number of bacterial cells, each having this modification in their genetics, and they will have the ability to then express the insulin gene, and then they will produce your insulin, which you can then extract and then bottle, purify, and give to diabetics, just like that. So this is just a sort of written version of what I'd gone through in the previous slide. So stage one The key things here, folks, the key sort of phrases is the identification of the DNA that you've got the required gene. We then, once we've got that, it's a cutting process, so cutting of the source chromosome using special enzymes, and that will release your gene. You're then going to extract the plasmid from the bacterial cell. You then open up a plasmid, or you cut it open using an enzyme. You then put the gene into the plasmid and you get the sealing process, and you get the insertion of the plasmid back into the bacterial host cell. After a period of time, growth and multiplication of the transformed bacterial cell occurs, and as a result you get many duplicates of the modified plasmid, and eventually you will be able to express the insulin, which can be then extracted and purified. Okay, so it's really important for the process of genetic engineering. You won't be asked ever to draw it from stage 1 right the way through to stage 8. It's really being able to identify and being able to describe and explain what's happening at each stage. So what you can do is you can use the following words and phrases to describe genetic engineering. For some, that will be a very straightforward process, maybe a wee bit tough in places. However, if that's too much of a leap to begin with, and you're thinking, wow, this is just too much biological words, too much phrases, what I suggest you do first is get a handle on the terminology before you start describing the process of genetic engineering. So for instance, what is a chromosome? What is a gene? What is insulin? What is a plasmid, etc. So if you go through all of those and be able to sort of get a handle on the terms, that might help you write your little paragraph on genetic engineering. Okay, once we have talked about the process of genetic engineering, we then talk about genetically modified organisms, and the definition of that was right at the start. A genetically modified organism is an organism whose genetic material is altered using genetic engineering techniques. What we've got there is a glowfish, the first genetically modified animal to be sold as a pet. So you will not find these up at the retail park in Kirkcaldy. But what I've got there is things to consider ethically. Do you think this is right? Would you buy a glowfish? If you would, what would you call it? Please glow better than what I used to call my fish when I was younger, and all it was Jaws, Fishy or Goldie. Try and be a wee bit more inventive. What would your glowfish be called? Or you might be in the camp where you think that's just completely wrong. That's not very nice. And ethically, you might think, no, I'm not in agreement with that. And that's okay. That's okay for you to think like that. It's your opinion. So genetically modified crops. These are crops that have had their genetic makeup altered, and there are 4 that we've looked at: golden rice, rapeseed, tomatoes and potatoes. And in the next few slides, it's just giving you just a wee bit more of a description of what's been genetically modified and why is that advantageous to us as humans. This is just a wee summary of the genetically modified crops, and what I've done is I've broken this down, given you a wee diagram just to try and visualise what each of these is like and what the modification has been. So golden rice is a genetically modified crop, and what has been genetically modified is that the rice produces something called beta carotene. Don't worry, you won't be asked to remember that. You will, however, maybe be asked to remember to be able to give an example of a genetically modified crop, and just be aware of what the advantage is. So beta carotene is converted into vitamin A, which can then be metabolized by the human body. So herbicide resistant, so most varieties most varieties available today of rapeseed are genetically modified to be herbicide resistant. Why is that a good thing? Well, this allows herbicides to be used to kill the weeds around the crop, but not the rapeseed itself. So what we're doing here is we're increasing the crop yield of the rapeseed. And rapeseed could be used to produce things like rapeseed oil. Tomatoes with extended shelf life. So a genetically modified tomato is a tomato that's had its genes modified, again using genetic engineering. And why would we want to do that? Think of extending the shelf life of food for consumers as well as the supermarket trade. So the first child of genetically modified food was the tomato. However, Flavour Saver, as it was called, never actually made it to market. So that is a genetic modification that would benefit humans in terms of food production. You get blight-resistant potatoes. If you're in my class, you know how much I love potatoes. Not. So even just to look at that slide of potatoes, and my stomach's going. But potato blight is caused by a fungus. So a genetically modified potato has had its genes modified so it can actually be resistant to that particular fungus. Again, if you're resistant to a certain fungus, that could increase your yield of that crop. So the benefits of genetically modified crops, like everything in biology, there's fors and against for most things. So GM crops and animals, because you can genetically modify animals as well, they can be higher yielding, better tasting potentially. They can increase food security, so think about the tomatoes and extending the shelf life, increasing your crop yield for potatoes, etc. So they can increase the food security for what is an increase in human population. They can have a positive impact on the environment because some of them might be resistant to certain diseases, for instance, so therefore you can decrease your pesticide use, and they can potentially play a part in the conservation of your water and energy. Issues, however, a lot of that comes down to safety and human health, environmental issues, people thinking that these genetically modified genes getting into the wild population. There is concern over the domination of food production by genetically modified companies in industrial countries. We've got ethical issues. People think that tampering with nature, is that right? And it's such a new technique that in the longer term we don't know what the potential problems could be. So some people, are on the fence of maybe being quite resistant for genetically modified crops. But again, that's entirely up to you to have an opinion on that. But as a true scientist, always look at both sides of the story scientifically and make up your own mind. As a process, in terms of genetic engineering, again, advantages and disadvantages. So advantages of genetic engineering as a whole, bacteria and yeast, they reproduce very, very rapidly. So you can make large quantities of a desired product in a very short space of time. So that could, swing people to say that that is a good thing. It's good for commercial uses. So, for instance, making detergents, for instance, and also medical advances. Just think of insulin production. However, as a process, the initial development of reprogramming microorganisms can be very, very expensive. So the setup costs are really, really expensive, and that might put a few companies off setting up. There is a possibility that genetically engineered microorganisms might be released into the environment, and people don't know what harm that may then cause. So like everything, you've got your advantages, you've got your disadvantages. Certain applications of genetic engineering, this kind of ties in with kind of the advantages. So the medical applications of using genetic engineering, you can use it to make insulin, growth hormone, your factor VIII. Other examples of genetic engineering, for instance, bacteria. We can use genetic detergents. You can make detergents, with bacteria to produce enzymes to remove difficult stains. For yeast, you can use genetic engineering to form yeast that produces alcohol with a high alcohol content and a low carbohydrate content. For cheese making, rennin can be transferred into yeast cells to manufacture vast quantities of a product. So these are all just kind of examples of both a medical and commercial use of how genetic engineering could be used in these two different industries. This is going to be a wee extension task, you can actually look at extreme genetic engineering, and we've got 4 examples there: the EnviroPig, venomous cabbage, glow-in-the-dark cats, and medicinal eggs. This is, if you're really interested in genetic engineering as a process, to get your teeth into looking at some of the extreme cases can actually be quite interesting. But again, that's up to your teacher whether they set you that as a piece of work or not, but a wee extension there. The last thing that we're going to look at is a term called excuse me, farming, and this the definition of it is the process where plants are genetically modified to produce medicine. So you're not looking at your bacteria, your yeast, we're actually looking at plants, and we are genetically modifying plants in particular to look at producing medicines. For farming, again, we're going to be looking at advantages, disadvantages, and plants such as corn, tobacco and rice can be used in farming, and they can be genetically modified through genetic engineering to produce proteins that could be used as medicines for humans. So the advantage of that, of producing such medicines in plants, is the product will be easier and cheaper to produce than is currently possible. And medically important products can be used in things like producing vaccines and hormones. So that is a good thing to do with farming. Bear in mind, I've not made a typo, it is farming PH, not farming with an F. For any of you who are sitting a bit sort of bamboozled when I'm talking about farming. But that is, think of pharmaceuticals, the pharmaceutical industry to use to make medicines. Against farming, well, farming is a form of genetic engineering, and people that are against genetic engineering, a lot of the arguments are pretty much the same. Although farm plants are not grown as food crops, there is the concern that altered plants might find their way into the food supply, cross-pollinate with conventional crops, and who knows what may happen. So some people are obviously against that process, that some of the genes, the farm genes, may enter into the food chain, and we don't know the impact that that would have on the human population. There are also some concerns that some of the chemicals produced by farm plants may also cause allergies. So again, it is very much yin and yang, some good things and some not so good things to do with farming. But overall, that is pretty much in a half an hour slot. That is the genetic engineering section in a nutshell. I hope you've found these slides useful, and any questions that you might have, please talk to your teacher through Teams, and hopefully this section will be really interesting for you, and good luck with all your tasks. Okay, all of us in the biology department are missing you all, and we really hope to see you all very, very soon. Okay, so take care and good luck with genetic engineering.